Okay, let's look at the investigation number 2. So at the investigation number 2, I already said it that we are going to look at the what at whether light it is required for photosynthesis or it's not required for the photosynthesis. So we're going to look whether do we need the light during photosynthesis or do not need it. So already we know that we need it, right? We've been told that we need light for the photosynthesis. And some people say, no, we have to do what? We have to prove it. You cannot just tell me that the light is required for the photosynthesis and I just agree. I have to do what? I have to do an experiment to prove it. How do we do this experiment? So the first thing is to do what? Is to do what we call a distancing. So we know we have to do the distancing so that the results that is formed, we know that in all our experiment we depend on the what? We depend on the starch test. Starch test. So the starch that will be formed, that will be testing now. It is not the what? It is not the starch that is formed before our investigation. It's the starch that is formed due to the what? Due to our investigation. We remove the old starch. Now we need to test for the new starch. So we have to perform the process of what? Of de-starching. After performing this process of de-starching, we can now begin with our experiment. Our experiment, we're gonna extract the plants now that have the what? That have this leaf. And in this leaf, we have to cover some portion of this leaf with the what? With aluminium foil. We cover this part with the aluminium foil. I'm gonna indicate it as label H part A, and there's this part that is not covered with aluminium foil. I'm gonna say it label is what? It part B. Then I'm gonna take my leaf now and place it at the what? At the sunny area for the what? For the 48 hours. Sunny area for the what? For 48 hours. So now as I place it at the sun area for the 48 hours, I will then remove the what? The foil. And after removing this foil, I'm gonna take my iodine solution and pour it at the part A and also at the part B. Remember the covered part, do not receive a what? Do not receive any sunlight during the time when I place it in the sun area. It doesn't receive any sunlight because it was covered. And part B received the sunlight because it was not covered. So there's absence of a light, and here there's a what? A presence of the light. So now I later perform the what? The starch test and to find whether there will be a presence of starch at part A or there will be a presence of starch at the part B. So now it means during my investigation, the independent variable is the variable that I'm changing. Now the variable that I'm changing is the what? Is the light. Is the present or the what? Of the absence of the light. It's our independent variable. But the dependent variable is the one that I'm recording. What am I recording? I'm recording the what? Whether the starch is formed or it's not what? It is not formed. So this is the what? This is our dependent variable. It is the starch. So for my experiment to be valid or for the fair test, I must make sure that other factor remain what? Remain constant. The factor that should remain constant now should be a what? Should be uh, the type of the plants. I should use the same type of the plants, and also I should use the what? The same amount of what? Of water. I know that water is what? Also required for the photosynthesis. I must not change it. The only variable that I should change is the what? Is the light. So water, I should not change it. It should remain the constant. And also the CO2 should remain the constant because it will interfere with the what? With our results. The factor that should be changed is only the what? It's only the light. I should only change the what. So I said in experiment there's a two part. There's the experimental part and also the what? The control part. Which one is my experimental part? So the part that is covered by the foil do not receive all the what? All the requirement. So it does not receive the sunlight because it was covered with the foil. So if it does not receive all the requirement, we call it a what? Experiment. And this part that was covered by the what? That was not covered. It received all the requirement. I call it a what? A control. It helped the light, it received the light. So I call it a what? a control, and it is used to what? It uses as a baseline to compare the results between the what? The experiment to the experiment. I use this to compare the results with the what? With this one of the experiment. And now let's look at our results. So in our results now, when we perform the iodine solution, the part that is covered, that do not receive any sunlight will remain brown, and the part that is not covered now will tend to be what? To be blue black. Will be black. The part that is not covered, that is covered, that is covered will tend to be what? To be brown. The part that is not covered will tend to be what? To be blue black. And so what does this mean? This now means that the covered part do not have a what? Do not have any starch. So if it does not have a starch, it means that now there will be no what? There will be no photosynthesis. And the covered part, no light. So the absence of the light, there's no photosynthesis. It means now light is required for the what? For the photosynthesis. We're done with this video. I'm gonna see you at the next video where we're gonna discuss another factor.